Okay, now we're going to cover the rest of Chapter 9 and again studying populations. So, a species can be characterized by its geographic range, where you find that particular species. As you can imagine, this varies a great deal from species to species and is influenced by many factors. And with some species, as shown here with monarch butterflies, for example, there are ones that migrate just like many birds, and so they have basically a winter range and a spring and summer range. And they basically migrate north in the summertime and then back south in the wintertime to Mexico. Um, with the monarchs, there's been concern about their populations because there's been some decline, and in particular, their habitats in Mexico are very restricted, and in some mountains around Mexico City, and habitat loss there has had a significant negative impact on their populations. So we talked about earlier, of course, populations can have very patchy distributions sometimes. Um, this image is of a particular species of plant called clematis, Clematis, or clematis, and it likes particular habitats. You can see in Missouri what are called these rocky outcroppings that are relatively dry, exposed habitats, but these plants seem to like it. So you can see again quite patchy distributions as we get down into smaller and smaller scales. Um, Again, even within this area right here that we see as a habitat, as you blow it up, again, we can see within it, it's relatively patchy as well. Again, common characteristics of species. Um, and so within a range, geographic range where you find a species, of course, they're not equally distributed across that range. Here's an example with kangaroos in Australia. So they basically are found throughout a good chunk of Australia, excluding those little areas there. But you can see within that range, there's a lot of variation in density. They're most dense down in this area, but in the green area is relatively low density. So clearly, for whatever the conditions are that kangaroos like, are quite good in this area and not so good in other areas. All right, dispersion. When we look at where we find a particular species and where the individuals are, they can be dispersed in different ways. These are sort of the three general types. You can have a regular dispersion. That is, they're just sort of relatively equally dispersed across the landscape. Here we have these um, shrubs in the desert southwest United States. Another good example there are penguins and their colonies. All their nests are equally distributed across the landscape. Dispersion can also be relatively random, no apparent pattern to it. And you can also have clumped distributions as well, as is shown with these trees in this wetlands area. There's only certain areas that are quite dry enough, or just dry enough for the trees, and the other areas are too wet. So it's a very clumped dispersion for that species. Okay, now, how can we go about estimating the size of populations? And typically we do have to estimate size because the absolute population size is the total number, but it's not always possible to get the total number because that can, of course, as you can imagine, take a lot of effort to do that. So often we get estimates of size, um, and sometimes when we get these estimates, we just look at them from year to year, and so we can also speak of the relative population size. And so we might say the population was relatively large in one year, but now our estimates are showing it's somewhat smaller. So we would say in that second year it has its relatively smaller population. Of course, that's an important thing to know for conservation biology and how species are doing. Okay, how can we estimate the population size? A common method is just what's called the area-based or quadrat method, where you establish a rectangle, a quadrat of a particular size, and you just count the number of individuals in that quadrat. And so, for example, if you were in, say, Percy Warner Park, that's a bad representation, but let's say it's Percy Warner Park, we wanted to find out how many particular types of trees out there. As you can imagine, it would be a lot of effort to go count all of them. But what you can do is establish various quadrats of the same size and get the number in each of those quadrats, and then you can extrapolate up and say, okay, we find X amount per 10 meters squared, and we can scale up to the whole park and get an estimate. Clearly, the size of your quadrat is determined by what you're sampling. If you're doing grasses or something, you'd have a very small one, but as I said with the other example, if you're doing trees, you would have to have much larger quadrats. You can also do what's called the distance or transect method, and this is where you just basically lay out a line across the landscape and move along that line, and depending on, again, what you're sampling, if it's, say, trees, it's just however many you can see on either side as you're walking along that transcript, you can get an estimate. Or if it's a smaller species, you would perhaps establish a certain distance to either side of that transect. And so then you would do several transcripts through the habitat. And again, you can get an estimate of the size of the population. Now, with things that move, in particular with animals, um, it can be a little trickier to use these other methods because, as we said, they are essentially moving. And so we have what we call here the mark recapture technique. And in this, you basically catch a bunch of the individuals, you mark them, let them go, and later you recapture them. And of the ones you recapture, a certain proportion of them will be marked. And from this, you can get an estimate of population size. And so it kind of works like this. All right, so N is what we're trying to estimate, okay? And M is the number of individuals that you have caught at one time and marked. C represents ones that are subsequently recaptured, okay? And R represents of those that are recaptured, which ones are marked. So for example, again, we're trying to estimate N, and so we can arrange this, rearrange this, and okay, M over R. And so if we have captured and marked, say, 20 individuals, okay, and we recapture in a subsequent um, capturing, say, a week later or so after we've let those 20 go, let's say we recapture 15 individuals, and of those 15, 4 are marked, okay? And again, this is all equal to n. When we do the math here, again, that gives us an estimate of n. And this formula works because these ratios should be the same. That is, when we capture and mark some, they represent a particular fraction of the total. When we let them go and capture them later, we should get the same ratio. Okay, and again, that should give us a pretty good estimate. We're going to practice this in class later. Okay, so here we go. Now, here's an example of a population study which had some particular relevance in the southwest United States. People were studying these populations of deer mice, and the populations would fluctuate somewhat, and which of course is perfectly natural. Um, but they noticed that there was a particular outbreak of this disease um, in people, and this disease is known as hantavirus, Hantavirus. And it was kind of surprising that there was this outbreak all of a sudden. Well, what they noticed was a relationship with the mouse population. So the mouse population had skyrocketed, which led to an outbreak of this hantavirus in people because the virus is found in mice and can be transferred to people. In particular, when the mice uh, defecate, when they poop, they have these sort of dryish pellets, and the virus is in there. When those pellets kind of break down, the dust can get in the air, and people can breathe in that and get the virus. Well, it was found that the outbreak again was due to a growth in the mouse population, which was related to the environmental conditions because there was a particularly wet year, and this led to an increase in the vegetation, which essentially allowed the mice to do particularly well that year, which meant they have lots of babies. So the next year, essentially, the population skyrocketed. Now, I'll leave it for you to think. What do you think would happen in the next year, though, once you have this large population of mice? Would it continue to stay that large, or might it shrink down some? Okay, now let's talk about the ecological niche. Ecological niche is essentially the range of conditions, both biotic and abiotic, that a species requires to survive, reproduce, and do its thing. Um, Here you can see we just have two conditions, moisture and temperature, and if we put them on a graph, we can see that there is a range of these conditions that are favorable for this hypothetical species. Um, Now, we can often think of this as what we call the fundamental niche. That is, that's where the species could live as long as all conditions are just ideal. But often the niche is somewhat more constrained. That is, there's a smaller realized niche. And that's because in this case, there are things other than just moisture and temperature that constrain that species. Um, Competition from other species, the amount of food that's available, a lot of the biotic conditions. That this we're just looking at some abiotic ones here. Um, as I'll mention in class, the ecological niche is sometimes described as a multi-dimensional hyperspace. That are many dimensions, many factors that constrain the species more than you can possibly put on a graph. Once you sort of have a good grasp on what a species niche is, you can then sort of do some estimating or guesstimating of where you might find that species out there in the environment where you think these conditions apply. And in Madagascar, that was done with geckos, and they were able to, again, study some populations of geckos, determine what the niche was, and figure out where on the island they should probably find other geckos. And the model worked pretty well. And that is about 80% of the time they were able to find geckos where they thought they would find them. Again, this is called niche modeling. All right, now last thing, we were talking about the sea urchins and the sea otters earlier, and a lot of research was done in that. Again, as I mentioned, in the late 1800s, early 1900s, the sea otters were hunted. They bounced back. That by the 1970s, the population was doing quite well, but then they began to decline again. Um, and the urchins made a comeback, and the kelp forest went down. In some areas where sea otters were reintroduced, you can see the arrow is time. This is the original time, and this is a later point in time. When the sea otters are reintroduced, the kelp do a lot better because, of course, or presumably the sea otters there eating the urchins. And this was in southern Alaska. In northern or more north and west Alaska, out in the Aleutian Islands, that long chain of islands that goes towards Asia, um, they didn't quite get the same results. Um, that is, the kelp did not come back quite as strong there. Um, Now, why did the sea otters decline from the 1970s on? It's thought that killer whales played a role and were significantly uh, de depleting the sea otter populations, which led to the more recent decline in them as opposed to the hunting that occurred earlier.